So, hello ladies and gentlemen. In my project I focused on the fungus known as Fusarium oxysporum. As you can see here, it is an ascomycete mold which grows ubiquitously in soil. You can find it almost anywhere on any continent, but some forms of Fusarium oxysporum are very dangerous because they pose a direct threat to plants. Fusarium oxysporum acts as a severe plant pathogen which is currently endangering crops of, for example, tomatoes in greenhouses, cucumbers and cucurbits in general, lettuce, chickpeas and other legumes, and most importantly bananas. Bananas are in imminent danger of being almost wiped out. The dominant banana cultivar, Cavendish, is a single clone of banana that you can get anywhere at a supermarket in the whole world, and it is critically in danger. When this fungus spreads to plantations in South America, then the world supplies of bananas will be very much in trouble. Alright, so as you can see, Fusarium oxysporum is a big threat to world food security, and in order to combat this fungus, I thought about using viruses. Using viruses of this fungus to cause disease in the fungus itself and make it cripple it and disable it from infecting plants. So the viruses infecting fungi are known as mycoviruses, and most of them have a dsRNA genome, that is double-stranded RNA. dsRNA occurs in nature only in viruses. Therefore, when an organism has a dsRNA present, we can be almost certain that they have a virus inside them that produces this dsRNA. They usually have a capsid or an envelope, and most of mycoviruses are harmless to their hosts, but some actually do cause trouble for the fungi that they infect. They may cause reduced pigmentation, stunted growth, and in our case, the most interesting trait, phenotype change, is hypovirulence. Hypovirulence means a decrease of virulence, the ability to infect a host, in our fungus. Mycoviruses are very hard to transmit by natural means. So, the objectives of this work were to assess the presence of mycoviruses in the species Fusarium oxysporum and to determine the phenotype changes that occur when a fungus is infected with the virus as opposed to a healthy fungus. Now, on to my methods. I used the double-stranded RNA as a marker of virus presence. As I've said, when a dsRNA is present in an organism, we can be sure that the organism is infected with a virus. So I used two independent methods of isolation of dsRNA. These methods are chemically independent. They rely on different chemical principles, and therefore I can be quite sure that the fungus does actually have dsRNA in it, and I don't have a false positive. I subsequently cleaved the nucleic acid extracts with nucleases. Therefore, I had pure dsRNA samples. This pure dsRNA was later subject to reverse transcription, PCR amplification, and molecular cloning into E. coli. These steps serve as an important intermediate in viral RNA sequencing, which will be carried out later on. So, I had a fungus with virus and without a virus. As you can see from the image that is not really there. And these fungi were compared in terms of growth rate on general medium, and morphology. That means how these fungi looked on petri dishes when growing, and also how they looked under the microscope. The most important test was carried out on plants. It was a test of fytopatogenity, the ability of the fungi to infect the plants and cause disease in them. As you can see from these images of Arabidopsis thaliana, a common model plant in plant biology, the control plants grew healthy and green. The plants infected with a fungal strain without a mycovirus were diseased. They quickly wilted and died as a result of Fusarium wilt. And the plants that were infected with fungi which did have a mycovirus inside them grew healthy and strong, just like the control plants. Therefore, we can see that the result of this test was that mycoviruses can actually produce hypovirulence in their fungal hosts and disable these fungi from infecting host plants. Which means that, in conclusion, I have confirmed presence of dsRNA mycoviruses in Fusarium oxysporum. I assessed the differences between a fungus culture which contained a mycovirus and which did not contain a mycovirus, where I found a very important piece of data, that is, a fungus with a mycovirus may possibly prevent the be unable to infect a plant actually in wild. Therefore, this work has a direct application in using these mycoviruses as a method of controlling fungi and therefore protecting vital crops worldwide. Here on the image you can see the principle of biocontrol. We would essentially manufacture a plant cure, if you will, for Fusarium oxysporum in the soil, present in the soil, which makes growing of plants impossible on the soil, and we would be able to remedy these soils and to restore farmland to farmers worldwide. In the future, I would like to perform more tests on actual plants, that is, repeat the phytopathogenity tests on more crops, such as tomatoes, lettuce, and peas, maybe even bananas. These crops are, that's what you're actually interested in when you're doing agriculture, so of course it's important to study those. Then I would like to test the transmission of the fungus. If one fungal culture can actually transmit the virus to another fungal culture, subsequently develop methods that can manifest this transition. And lastly, I would like to finish the sequencing of the viral dsRNA obtained in the first few steps, which would enable us to taxonomically place the virus and to describe it as a new virus species. Thank you for your attention, and ještě tam budou zdroje.